Hello, everyone. My name is Edward Carlsson, and I'm a industry PhD student at SLU and the Forestry Research Institute of Sweden. And as we've been hearing today, we all understand that using trees and wood biomass can be used for a lot of different applications, such as construction, wood biofuels, and advanced materials. And we also heard earlier today that Norway spruce specifically is a very important species in Sweden, being one of the two dominant tree species in abundance and for the industry. But how do we actually get these trees that we need for all these products? What makes a tree grow? Simply put, it's a combination of environment and genetics. And we can control part of the environment through our management practices. But to improve the genetic side, we can make use of tree breeding. However, breeding for Norway spruce is not that easy, because, as we've also been hearing today, Norway spruce takes a very, very long time to complete one cycle of breeding. We would have testing, selecting, and crossing. And in the end, you would have genetically improved trees that have higher survivability, better growth, and better adaptation to future climates. However, with genomic selection, this method proposes to decrease this cycle time by making selection at the seedling stage using DNA markers and the measurements from the previous cycle. So here we're using tens of thousands of DNA markers across the entire genome, not just one or two genes. And we use this to build a prediction model that can rank the trees within the next generation before they've reached this mature stage. The question is, how accurate can such a model be when we try to select a tree which we cannot yet measure? And in my study, I've tried to answer this question using a real-life data set with two generations, which is this green color here. And the accuracy is not that great. However, you have to consider that also there's a huge gain in reduced cycle time. Then I used a simulation to replicate this study with the same data and the same analysis, and saw that simulation agreed fairly well with reality. And this made me confident to take this simulation tool and ask questions about the future of genomic selection in Norway spruce. And we can see that in the future, it will actually increase in accuracy, largely due to building up structures in the genome that will help improve genomic merit. Thank you.